The pathway from the outer ear to the inner ear is remarkable in its ability to precisely process sounds from the very softest to the very loudest, and to distinguish very small changes in the frequency of sound, what you call the pitch. Humans can discern a difference in frequency of just 0.1%. This means that humans can tell the difference between sounds at frequencies of 1000 Hz to 1001 Hz. That is just 1 Hz difference, we can tell the difference. We use this ability in your music. You know that? No. The outer ear is composed of two parts. The pinna is the outside portion of the ear and is composed of skin and cartilage. The second part is called the ear canal, called the external auditory canal. The pinna, with its twists and folds, serves to enhance high frequency sounds and to focus sound waves into middle and inner portions of the ear. Pinna also helps us determine the direction from which a sound originates. However, the greatest asset in judging the location of a sound is having two ears, because one ear is closer to the source of the sound than the other. The brain detects slight differences in the times and intensities of the arriving signals. This allows the brain to approximate the sound's location. Interestingly, the position and orientation of the pinna at the side of the head help reduce sounds that originate behind us. This helps us hear sounds that originate in the direction we are looking and reduce distracting background noises. Some people believe that the size of the ear is an indication of the organism's hearing ability. That is, larger the ear, the better the ability to hear. This is a misconception. This misperception doesn't take into account the internal structures of the ear that process the sound vibrations. A large pinna may serve a function that is unrelated to hearing. For example, the external ear of an African elephant is filled with some blood vessels that help the animal dissipate excess heat. The external ear may be specialized in other ways as well. Cat owners, for example, have undoubtedly observed the rather dramatic movement of their pet's pinnae as the animal attempts to locate the source of a sound. Now, the ear canal. It acts as an amplifier for sound frequencies between 3000 and 4000 Hz. The ear canal is about 2.5 centimeter long and leads to the tympanic membrane or eardrum of the middle ear. The outer 2/3 of the canal contains glands that secrete a wax-like substance, the earwax, along with hairs that are present, serves to keep the dust, the insects and other foreign material from going deeper into the ear. It has a protective role. It also helps maintain a constant humidity and temperature for the middle ear. Individuals should not attempt to remove earwax, since the secretion will work itself out of the canal naturally in most cases. To avoid damage, it should be removed by a medical professional. Hearing researchers strongly concur with the truth of the adage, put nothing smaller than your elbow into your ear. What does it mean? Don't tamper with your ear, that's it. In addition to its protective function, the ear canal acts as an amplifier for sound frequencies between 3000 and 4000 Hz. The middle ear, the tympanic membrane, or what you call the eardrum, separates the outer ear from the middle ear. It is continuously growing structure, which means that damage to the membrane can generally be repaired. The membrane is circular in shape. The elastic properties of the tympanic membrane allow it to vibrate in response to sound waves. Vibrations from the tympanic membrane tend to focus near the center of the structure. From there, vibrations are transferred to the three bones in the middle ear. The first is malleus, the second is the incus, malleus, incus, stapes, collectively called the ossicles. So the vibrations are first transferred to malleus, then to the incus, and then to the stapes. Then each of these vibrate, in succession. And when stapes vibrates, the stapes is sitting upon an opening in the bony wall called the oval window that separates the middle ear from the inner ear. The elegance of the middle ear system lies in its ability to greatly amplify the sound vibrations before they enter the inner ear. Amplification occurs in part because the tympanic membrane is 15 to 30 times larger than the oval window. This size difference allows the force from the initial movement of the tympanic membrane to be concentrated as this energy transfers to the inner ear. The ossicles are the smallest bones in our body. The 3 bones are smaller than an orange seed. You can imagine how small it is. The malleus reaches an average length of 8 millimeters, incus 9 millimeters, and the stapes is the smallest, 3 millimeters. These bones also are referred to informally as the hammer, anvil, and stirrup because of their shape, respectively. Middle ear is an air-filled space. It is connected to the back of the throat by a small tube called the eustachian tube, which allows the air in the middle ear space to be refreshed periodically. The eustachian tube can become blocked by infection, and fluid may fill the middle ear space. Changes in air pressure can also affect the tympanic membrane, resulting in the ear popping phenomenon experienced by people who fly in airplanes or drive other mountain roads. The membrane may bend in response to altered air pressure and then pop back to its original position when the eustachian tube opens and internal and external air pressures are equalized. The process of converting vibrational energy of sound into nerve impulses, transduction. The inner ear, 2 interconnected parts that form a system of small cavities and passageways make up the inner ear. One part is the vestibular system, responsible for helping maintaining the balance. Second is the cochlea, a coiled cavity 35 millimeters long. The human cochlea makes 2 turns. It is shaped like a spiral seashell or snail shell and is the hearing portion of the inner ear. It is responsible for converting the vibrational energy produced by the middle ear into a nerve impulse. So the vibrational energy is converted into an electrical energy in this organ that will travel to the brain. The process of converting energy from one form into another is called the transduction, because the brain is incapable of interpreting the information in the vibrational energy of a sound source. Transduction is a critical process, providing information to the brain in a form that it can process. That is why only the electrical impulse can be grasped by the brain. So always the Kundalini has to be Saudamini Sannipa, just like an electrical thing as we discussed earlier. An uncoiled cochlea to the right of the oval and round windows. You can just see the structure divided into an upper chamber, which is called the scala vestibulae or vestibular canal, the lower chamber, the scala tympanae or the tympanic canal. These are seen most easily if the cochlea is represented as uncoiled, but it is not an uncoiled structure. Both the upper and lower chambers are filled with a fluid called the perilymph, which is nearly identical to the spinal fluid in our brain and spinal cord. It's a very similar fluid. The stapes vibrates against the oval window, creating vibrations in this fluid that are transmitted as pressure waves all the way through the cochlea. As represented by the arrows, these waves move from the upper chamber to the lower chamber to the round window. The round window allows the release of the hydraulic pressure caused by vibration of the stapes in the oval window. Additionally, the diameter of the chambers decreases from base, that is closest to the windows, to the apex. You can see the diagrammatic representation of and movement of vibrational energy here. You can see the stapes sitting on the oval window, the upper chamber, the vibrations carried from the upper chamber to the middle chamber, then to the lower chamber. The round window membrane you can see there. Then you can actually see how it is transmitted from the 3 bones into the inner ear cochlea and how it reaches the from the middle ear to the inner ear. So upper and lower chambers are separated from one another by cochlear duct. Cochlear duct is separated from lower chamber by basilar membrane, and it is filled with endolymph, a fluid similar to that found within the cells also. sitting on the basilar membrane is the highly sensitive organ of hearing called the organ of Corti. You can see that organ here in the picture, named after Alfonso Corti, an Italian anatomist who discovered it in 1800. The relationships between the basilar membrane and the organ of Corti are depicted here. Now, the hair cells of the organ of Corti are the specialized receptor cells of hearing. Under a microscope, these cells appear as elongated oval cells with hair-like extensions called the stereocilia, waving at one end. Like microphones, the microphones you use in your day-to-day life, hair cells ultimately translate or transduce mechanical vibrations occurring in the outer, middle and inner ear into electrical impulses. These nerve impulses are then relayed to the brain via the auditory nerve. There are actually two types of hair cells. The inner hair cells are arranged in a single row along the full length of the organ of Corti. There are about 3,500 of them in total. The outer hair cells run the full length of the organ of Corti but are arranged in three parallel rows. They are nearly 4 times more outer hair cells than the inner hair cells, about 12,000 per year. The inner hair cells contact nearly all of the nerve fibers of the auditory nerve that transmits information to the brain. The outer hair cells primarily contact nerve fibers that carry information from the brain. Hair cells are quite sensitive to stimulation by slight sounds and also are extremely rapid in their responses and communication with auditory neurons. Hair cells, for example, respond 1,000 times faster to stimulation than do visual receptor cells. The key to their sensitivity lies in part with their structure. The membrane-bound hair-like structures that give hair cells their name, stereocilia, extend from the cell tops and are embedded in an overhanging sheet of cells called tectorial membrane. Each hair cell have 100 stereocilia. In a resting state, these stereocilia lean on one another and have the overall appearance of a conical bundle. Organ of Corti, you can see here the picture with the relationship to the hair bundles to the adjacent membrane. You can see that outer hair cell, you can see the stereocilia and the structure of the inner hair cell, like that. How it is connected, the entire thing is connected to the auditory nerve. How hair cell functions to transduce the mechanical vibrations of sound. The bodies of hair cells sit on top of the basilar membrane. The stereocilia of hair cells connect the body of the cell with the tectorial membrane. So pressure waves in the cochlea move the basilar membrane and cause the stereocilia to move. This movement initiates biochemical events in the cells that result in the generation of electrical signals. Sound is mapped to different parts of the cochlea according to frequency. The tones of different frequencies cause vibrations of maximum amplitude along the length of the cochlea. The base close to the stapes is stiff and narrow, responds more to high frequency or high pitched sounds. The apex far from the stapes is broad and responds more to low frequency sounds. You can see the diagram here showing the frequency of the hertz. The base 0.04 millimeter wide, 20,000, and at the apex 0.50 millimeter wide, only it reduces to 200. And transmission to the brain, extending from the organ of Corti, are 30,000 to 40,000 nerve fibers that form the auditory nerve. The number of fibers required to carry a sound signal may give the brain a measure of the sound's intensity. The fibers of the auditory nerve proceed a short distance to the brainstem. From there, the fibers extend to the midbrain and then to the auditory cortex, which is located in the temporal lobe of the brain. Through mechanisms that remain unknown, the brain interprets the electrochemical information it receives, thus allowing us to perceive sounds as having varying loudness and pitch. Brain recognizes and interprets sounds in our environment through a sequence of events called auditory processing. A disorder known as auditory processing disorder came to prominence in the 1970s. In it, something interferes with the brain's ability to process or interpret the information about sound. Although hearing seems to be normal. Hearing is normal, but the processing has gone wrong. Children with APD typically have normal hearing and intelligence. Symptoms of APD are having difficulty in paying attention and remembering information presented orally. Poor listening skills, difficulty carrying out multi-step directions, poor spelling, vocabulary is poor, reading comprehension skills are poor, difficulty in processing information, low academic performance, behavioral problems, language difficulties, tendency to confuse syllable sequences, difficulty developing vocabulary and understanding the language. APD is sometimes called a word deafness. The Shruti is okay, but there is word deafness. The language is not there. The children with the disorder may not recognize the subtle differences between the sounds in words. That is the problem. The subtle differences are lost. For example, children with APD may hear the sentence, "Tell me how a couch and a chair are alike." They will hear the sentences, "Tell me how a cow and a hare are alike." So, that is simply an example only. It means that they doesn't understand what the subtle things in it. That is only a Western man's humorous way of putting things. What causes this apparent deficiency or slowing in the brain's ability to process auditory information? Researchers do not know. Auditory processing is a learned function. And if something interferes with the brain's training, the result may be a deficit in the capacity to process sound. Sound direction is localized by virtue of our having 2 ears and our ability to use different parts of the auditory system to process distinct aspects of incoming directional information.